Color correction. Reading Lumetri scope's instruments. It's time to talk a little bit about color and its nature. How's the process of working with light built? How is it read and how is it interpreted? Firstly, we consider how it works in Premiere. There is a separate panel called Color at the top that is basically similar to the standard editing panel, but here the Lumetri Color panel appears on the side. In addition to the fact that this panel appears, the selection follows playhead is also activated. When we were working in editing, it would be not active. When we played the video, the playhead would move separately from them. This checkbox is activated in color mode, and if we play the clip and then put it on pause, it will be immediately highlighted under playhead. I move the playhead to the other side, another clip is immediately highlighted. Why is this done? How does the Lumetri color panel work? In fact, as soon as we apply an effect and change something in the Lumetri color panel, we immediately have this effect in Effect Controls. Here we have the Project panel, the Effects tab, the Video Effects folder, Color Correction, where Lumetri color is located. It has absolutely all the same settings as on the Lumetri color panel, but they are more conveniently laid out on this panel. If we move an effect from the Effects folder to Effect Controls, we will see it in this form: menu, submenu, etc. It's not as convenient and clear as it done on the Lumetri color panel, and as soon as we move the playhead, the clip is highlighted and any parameter changes, we immediately have Lumetri color in Effect Controls, and all further changes are reflected within this effect. As soon as we activated the color panel, we got the Lumetri Scopes panel. It appeared a couple of years ago. Before that, there were other tools. This panel works in real time. The old panel only worked with paused videos. How to work with it, how to read it, how to understand it. Look, we have several tools. Now I will turn them on by right-clicking on Lumetri Scopes. All these tools exist so that we can analyze the picture not only with our eyes. The human eye is often blurred and makes mistakes when working with color for a long time. This applies even to such an elementary thing as white balance. Why can't we see the difference between daylight and electric light? Because our eye adapts instantly, while the camera matrix doesn't work on its own and can't quickly catch light like a person. Our perception of color in the frame is influenced by the electric light in the room, the monitor settings, and any other factors. Therefore, we need a single objective scale that will help us distinguish colors and give other detailed information. These tools exist for this purpose. We won't consider all the tools because they are generally similar and duplicate each other in some sense. I will tell you about the ones that I mostly use. Let's look at what tools we have. Let's start with Parade RGB and Waveform RGB that we can consider simultaneously. You can know that they are similar. How to read them? The tool on the right is called Parade, and on the left is Waveform. The position of a frame is displayed from left to right. This is the left edge of the frame, and this is the right edge of the frame. That's it there. What can we see in the video? Here we have a lot of dark, here we have a flash of light, and here we have some middle shades. There are pixels arranged by brightness from the bottom to the top. The brightest pixel is at the top, and the darkest one is at the bottom. Thus, the hand looks like this spot here. The bright spot is right here. Well, then we can note that the frame is dominated by yellow warm shades. So the red graph is slightly higher. We see the same RGB parade on the RGB waveform, but on the one graph. We see separate spectra of red, yellow and green on the RGB parade. For the demonstration, I will create a gradient that will also help me. The gradient is prepared. So we see here, we have a transition, on the left black, on the right white. You see, If we don't have a color as such, all three lines will match in RGB parade. As soon as one of them rises above the others, it means that this color begins to dominate. Or for example, we increase the green color, so it rises on RGB devices, and magenta falls. Magenta is a combined channel of red and blue. RGB parade and waveform are about the brightness of the color. What can we talk about next? Here we have a little yellow. There is quite a contrasting picture. We have absolutely black and absolutely white. In general, a large number of pixels in the lower part. That indicates that the image is quite dark. Let's move on. Here we see an overexposed image, where there are many medium and many bright colors. There is no only black color that makes the picture look pale and soapy. Actually, but most cameras, except for usual cameras, phones, etc., always shoot a picture with a slightly larger spectrum that they show. Therefore, if you disable this checkbox, you can see that you can extract some detailed information from the black and from the extra light. In fact, there is nothing here, because there is the same picture with different settings. For understanding, if the picture is darker, more pixels are at the bottom. If the picture is lighter, more pixels are at the top. Here we have too much green shade. We see that the blue color rises above the others. The next picture, if we put it butt to butt with this picture, is underexposed, and this one, on the contrary, needs to be made a little darker, because these wheels can be made black, but we don't have black here, and here we can whiten a little, raise the highlights to appear lighter colors. We make it lighter and more contrasting. We see all this information on the devices. We'll periodically need to look at these devices to see if we have got extra lights and get extra shadows, in order not to lose details from the bottom edge. In principle, it's clear with these devices, because they are about the same thing. Next we have a histogram. We can imagine that these pixels are in the form of a sand, and then we turn this graph over, and all the pixels fall apart. That's about how we get the histogram then. If you place all the pixels to this edge to get hues, you will get our histogram. Therefore, I rarely use it, because it's less visual and has less information. It can be used to understand the distribution of pixels by brightness, which are larger and which are smaller, and of the specific white of the image. Here it's darker, here it's lighter. But we can still see in these devices where they are located. These three graphs are responsible for the brightness of our frame. Dectoscope YUV is about light saturation. Brightness and saturation are two different parameters that also describe light. The further the pixel is from the center point, the more saturated it is. Accordingly, we can see that the points refer to the color in which direction they go. Here is essentially a standard color circle. For example, we can see from this frame the central splash is directed in this direction. Everything else is not saturated, because the border of a frame saturation limit is located on the outer hexagon through these cubes. Everything that goes beyond this limit in terms of saturation turns into defect. The effect is the same as that of extra light, but in this case it's oversaturation. The light can be so bright that the detail in this place simply disappears. We also should avoid it. We need the saturation to be somewhere within these limits. Let's see how the cloud of saturation spread when we are changing the parameters, because the saturation is almost everywhere. We can see as we are increasing the saturation as this cloud is spreading. And if we lower it, the picture will become black and white. This is almost close to defect. So, the picture is heavy for perception, and TV channels will block this and return it for processing, because the saturation level is too high. This is all about the devices. Next, we consider the specific blocks.